How did people determine biological relationships before modern technology? Today we have incredibly precise DNA testing, but this wasn't invented until 1984. Before that milestone, throughout the vast span of human history, how did our ancestors figure out a biological relationship? The very first approach, used for the majority of human existence, was physical observation. This was straightforward. People looked for distinctive traits, like hair color, eye color, or prominent birthmarks. But the flaw here is obvious. Superficial traits like birthmarks or moles are rarely hereditary, and the genetics behind hair and eye color are incredibly complex. Furthermore, if everyone in a community shares the same ethnic background, meaning they belong to the same racial or geographical group, relying on hair or eye color is useless. If everyone around you looks similar, physical observation fails to prove a thing. The second method is a fascinating one you have probably seen in historical movies: blood mixing. In ancient royal dramas, if a king suspected the queen of an affair, royal doctors would mix the child's blood with the father's in a bowl of water. If the blood cells clumped together, a phenomenon modern medicine calls agglutination, they believed it proved there was no biological connection. Today, we know genetics is far more complex. A child inherits 50% of their genes from the mother, and our blood type is determined by two alleles, which are alternative forms of a gene. For example, a father with type of blood could have the genotype, the specific genetic makeup, of OA or A. A mother with type B blood could have a genotype of OB or BB. If an OA father and an OB mother have a child, there are 4 possible genetic outcomes. Only in the single scenario where the child inherits the exact O, a genotype from the father, will the blood mix smoothly without clumping. In the other 3 scenarios, a massive 75% chance, the blood will clump, even though the father and child are 100% biologically related. This flawed test undoubtedly led to countless wrongful executions throughout history. By the 1920s, medical science advanced. While doctors still couldn't use blood types to pinpoint exactly who a father was, they could use them for exclusion, meaning they could definitively rule someone out. For instance, if a child has type O blood, their genotype must be OO. This means the biological father must carry at least one O allele, meaning his genotype could be OA or OB. If a suspected father had a genotype like a A, BB, or a B, he could be 100% ruled out. Later, science advanced further with a method that was over 90% accurate, HLA testing, which stands for human leukocyte antigen testing. Think of these antigens as a biological ID card that every cell must present to our immune system. If the immune system sees a foreign ID card, it destroys that cell. Just like a real ID card, these antigens have incredible specificity, meaning they are highly unique to an individual. Only immediate family members share specific allele codes. However, this method had major drawbacks. It required a massive amount of white blood cells, it was highly expensive, and if 2 unrelated people shared a very close ethnic background, their halocodes could match closely enough to skew the results. This brings us to the ultimate breakthrough we still use today, DNA testing, which revolutionized forensics in 1984. Today we can trace ancestry not just back 1 generation, but across thousands of years. You have probably noticed that modern DNA samples are usually collected using a simple cheek swab. Why the cheek? First, the cells in our oral mucosa have an incredibly high turnover rate, meaning they replace themselves very quickly. Swabbing for just 30 seconds collects thousands of fresh cells. Second, because the sample comes from inside the mouth, it is shielded from environmental contamination and less likely to pick up someone else's DNA. Finally, it is completely non-invasive, meaning it requires no needles and causes no pain. Could we use blood instead? Yes, but blood collection is invasive and painful. More importantly, as you learn in physiology, the science of how living organisms function, mature red blood cells lose their nuclei as they develop. Because they have no nucleus, they contain no DNA. To run a DNA test on blood, scientists have to complete the extra difficult step of isolating the white blood cells, which are the only blood cells that still keep their nuclei. Beyond a simple cheek swab, there are a few other common ways to collect a viable DNA sample. Do you know what they are? If you can guess these alternative methods, let me know.